Module 16, the reproductive systems. Spermatogenesis, development of sperm. The process of meiosis produces different products for males and females, so let's first discuss the development of sperm in males. The process happens in the walls of the seminiferous tubules of the testes. Look at this image. This section represents tissue between the seminiferous tubules, and this is the area of the edge of the seminiferous tubules. Diploid cells, called spermatogonia, first undergo mitosis. One of the two cells that results from this mitosis remains a spermatogonium, but the other becomes a primary spermatocyte. This is the cell that will undergo meiosis. Now, a primary spermatocyte still has 46 replicated chromosomes. It will undergo meiosis I, forming two secondary spermatocytes. Each of these cells has only one chromosome from each homologous pair. Each chromosome is still replicated, though, the copies attached at a centromere forming an X shape. Therefore, there are 23 replicated chromosomes in each secondary spermatocyte. Next, these cells undergo meiosis II in order to separate the replicates. The result is 4 spermatids, each containing 23 non-replicated chromosomes. This means that each spermatid only has one allele for each gene. You can see here in the diagram that the process of mitosis, meiosis I, and meiosis II happens as the developing cells are pushed along from the edge of the seminiferous tubules to the lumen in the center. At this point, the spermatids develop notable features and then enter the lumen of the seminiferous tubules so they can be moved into the epididymis and mature completely in order to be able to swim. The duration from the beginning of mitosis in the seminiferous tubule to the mature sperm cell lasts about 70 days. Once mature, a sperm cell can live in the male for many weeks. It still will not move on its own, rather being moved by smooth muscle contractions of the surrounding ducts. Outside the body, sperm are able to swim and can live for a few days within the female. I mentioned that after meiosis II, the spermatids develop notable features before moving into the lumen of the seminiferous tubules. There are 3 sections to a sperm cell: the head, the midpiece, and the flagellum. At the head is a cap called the acrosome. It is full of digestive enzymes necessary for eating away at the covering of the ovum. During normal fertilization, several sperm will attempt to fertilize a single ovum, using the enzymes in the acrosomes to break down the ovum covering. The first one to make it through will fertilize the egg. The nucleus, containing 23 chromosomes, is also in the head. The midpiece of the cell is where the mitochondria are. You see, it takes a lot of energy for the cell to swim, so the sperm need many mitochondria to provide that energy. The flagellum, of course, is for movement, so the sperm can swim toward the ovum. Can you see the specific design of this cell? It is produced for one thing: to fertilize an ovum. Thus, it has to be very lightweight and mobile. So besides mitochondria for power, you won't find lots of other organelles in this cell. Over half of the cell is flagellum. The mitochondria provide the energy needed to propel the cell, which basically is just carrying the cargo of DNA. The only other major structure is the acrosome that is to help the sperm drill into the ovum for fertilization. Upon fertilization, only the head makes it into the ovum, so a sperm cell only contributes its chromosomes. Now, sperm cells are the smallest cells in a human body. Tens of millions are necessary to be released in order for the possibility of fertilization to occur. If half the number, or about 40 million, are produced, then that man is said to have a low sperm count, making it more difficult for fertilization. If there is only about a fourth of the normal number produced, then that person is considered infertile. Now, in the diagram, there are 2 cell types that I have not introduced. They help in the process of spermatogenesis. The first helper cells are sustentacular cells, which help the developing spermatocytes by forming a liquid-tight blood-testis barrier around them while they're inside the seminiferous tubules. This is important because during their development, the sperm can create antigens that the body would attack. Therefore, the blood-testis barrier helps to provide an envelope of protection around the developing sperm so they cannot be seen by the body. However, this seals the developing sperm off from the blood supply with its nutrients and oxygen, so the sustentacular cells give them what they need. These cells also produce a hormone called inhibin. This hormone is involved in a series of secretions to help control the spermatogenesis rate. The second set of cells here are called interstitial cells, or Leydig cells. They are found outside the seminiferous tubules in the testes, and they secrete testosterone. Module 16, the reproductive systems. Experiment 16.1, spermatogenesis and sperm. In this lab, we're going to take a look at the seminiferous tubules and sperm, and then compare those sperm cells to other cells so we can get an idea of the size comparison. So we're going to first start with a slide of human testes, and let's put that under the microscope at 40 power magnification, and center in and focus. All right. So what we're looking at is you're going to see kind of a lot of ovals that are surrounded by some cells, like an outline of cells. Those are cross-sections of the seminiferous tubules where the sperm are produced. Go ahead and center one of those and increase your magnification to 100 power. All right, and we're focusing on some of those now. At this magnification, you can see that some of those ovals have kind of a stringy tissue in the center. Center on one of those stringy areas and again increase the magnification to 400 power. So I'm going to center over that stringy material to 400 power magnification and focus. And now we can get a good look at a cross-section of the seminiferous tubules. All right, looking at this oval shape, we want to start with the edge at first. You should see a line of distinct tissue surrounding it, and that is the basement membrane. In between the basement membrane of the two different ovals is interstitial tissue. When you make an illustration of this, you're going to want to label the interstitial tissue and the basement membrane. Now, there are lots of cells in the basement membrane and interstitial tissues, and most of them are fibroblasts. A lot of them are. And the cells with a lot of space between the cell membrane and the nucleus are the interstitial cells. Directly beneath or underneath the basement membrane, you're going to find spermatogonia. And under the spermatogonia, you're going to see primary and secondary spermatocytes. And these are kind of circular shaped cells. In this area, you're going to see some cells with some irregularly shaped nuclei too. You may have to look around a little bit to find those, but these are the sustentacular cells. In the center of this cross-section, you're going to see tiny little filaments. Those filaments are actually the tails of developing sperm. So at this point, you want to make an illustration of what you're looking at 400 power magnification, identifying all of the things that we just discussed. And once you've completed that, we're going to look at our next slide. So I'm going to take my slide off of the stage, and we're going to look at human spermatozoa now at 40 power magnification, and we want to focus in. Now your text says to look for a pink line of material. From one slide to another, you're going to see variations, and you may have noticed that as you've gone through this course, that some things are slightly different in color or different shapes as to what I've been describing. The slide that we're going to be observing for our spermatozoa actually has bluish, purplish blue tint to it instead of pink. So just be aware that the color is going to be a little bit different. So we're at 40 power magnification, and your first thing you're going to notice is that sperm cells are very small, very small. Now we want to focus on 100 power magnification, and you should be able to see tiny little splotches. Again, in my case, they're bluish splotches with filaments, and those are, again, the sperm cells. We're at 100 power magnification, and notice how tiny they are. Go ahead and center some of those and increase your magnification to 400 power, and focus in and center some of those again. Now take a look at the size of those cells. We're at 400 power magnifications. They look like a tiny dot with a filament coming off of them. All right, the dot is the head of the sperm and the filament is the tail of the sperm. And these are very tiny, tiny cells. Make an illustration of what you're looking at 400 power magnification, identifying the head and the filament. And again, take note of the size within the field of view that you have right now, because in just a second we're going to look at one more slide. All right, once you have made your illustration, Let's move the magnification back to 40 and remove this slide, keeping in mind the size of those cells you were just looking at. And we're going to take a look at our human blood smear cell, or slide. I'm putting that at 400 or 40 power magnification, and I'm focusing. These are blood cells, and we remember erythrocytes are very tiny to start with. Go ahead and increase the magnification to 100 power and focus again. All right, and now let's move up to 400 power magnification. And hopefully the other slide was fresh in your mind. And you can see that a red blood cell is much larger than a sperm cell. Go ahead and make an illustration of a couple of red blood cells next to the same illustration of sperm cells that you made and identify that just for size comparison. So although erythrocytes are very small, sperm cells are much tinier. And I hope that you got a better understanding of that by doing this lab. So once you've completed that, go ahead and clean everything up and put it away.